Hello and welcome to Mscdfsm Biochemistry, and we are covering here lipids and proteins part 2. So today we are taking topic neutral fats. They are the ester of fatty acid and glycerol, and they are insoluble in water and soluble in ether. They are good solvents. They are bland, colorless, neutral in reaction. When one molecule of fatty acid bind with glycerol, they form monoacyl glycerol. When two molecules of fatty acid combine with glycerol, they form diacyl glycerol. And when three molecules of fatty acid combine with glycerol, they form triacyl glycerol. What are phospholipids? Phospholipids are heterogeneous compounds widely distributed in animal in cell membrane. Phospholipids are further classified on the basis of alcohol present. Glycerophosphates, in which glycerol is alcohol. Phosphoinositides, in which alcohol present is inositol. Phosphos Phosphosphingosides, in which sphingosine is the alcohol. Glycerophosphatides, they are the ester of fatty acid with glycerol containing esterified phosphoric acid and nitrogen base. Brain, nervous tissue, liver, kidney, etc. has large quantity of phospholipids. What are the structure of phospholipid? First, phosphotiric acid is formed. In this, the carbon number 3 phosphate group is present. When two fatty acids are present at carbon 1 and carbon 2, this is the simplest form. It is similar to triacyl glyceride except that carbon 3 of glycerol backbone is esterified to phosphoric acid. Structure of phospholipids, it is very important. Whenever you have question about phospholipid, what is this, you have to make this structure. If you are making the structure, then it is very good to score good numbers. So this is the structure and it's very easy if you check it deeply. And then the function of phospholipid, they increase the rate of fatty acid oxidation, they help in blood clotting, they form membrane structure, they act as inorganic ion carrier across membrane. Example, lecithin, cephalin, cardiolipin. Lecithin is found in brain, lung and spleen. So structure of lecithin, if you ask about lecithin in 2 numbers, 3 numbers, then you can again make the structure of lecithin. Yeah. What are steroids? Steroids are the group of compounds that are associated with fats. They are soluble in fat but resistant to sodium hydroxide. What is the structure of steroids? It contains 3 cyclohexane ring. You can see A, B, C. This is cyclohexane ring and D is pentane. So it has 3 cyclohexane ring and 1 cyclopentane ring. The parent compound of steroid is called cyclopentanoperhydrophenanthrene that has 2 methyl chain at position number 10 and 13. Type of steroids. What are the types of steroids? One is sterol. These are the steroids that contain alcoholic group at C3 position and side chain of 8 to 10 carbon atom at C17 are called sterols. Examples are cholesterol and ergosterols. We all know what is cholesterol. Cholesterol are the alcohol with cyclopentanoephenanthrene ring system. This is also found in testosterone, cortisol and progesterone. These are all hormones. Cholesterol is considered as alcohol as it has OH group in position number 3 and it is found in brain, nervous tissue, adrenal and skin. So that is why we always say that cholesterol is important. It is not always bad, but if it is present in skin, that skin has its shiny structure, lustre. Then we will see, because it is also a good question, what is cholesterol, like structure of cholesterol. So you can see this structure and it is also very simple. It looks complicated, but if you see closely 1, 2, 3 and 5 a cyclohexane, and then one pentane, and after that it's a side chain. So you just have to put the numbers here. So this is the cholesterol structure. And next is, what is ergosterol? This is found in yeast, mold. Sterol of vegetable origin are called phytosterol. They have same structure as cholesterol but differ in side chain attached to the carbon number 17. Phytosterol such as stigmasterol from soybean as they lower blood cholesterol. Second, other type of sterols are bile acid, glycocholic and taurocholic are the examples. Another type is hormone. Examples are testosterone, corticosterone. Vitamins, vitamin D2 and D3 are examples. Cardiac, glycosides. Example is tretopentine and saponin, digitonin. What are ecosinoids? Ecosinoids are the local hormones formed during body healing. These are derived from polyunsaturated fatty acid, eicosanoic acid. They are produced from arachidonic acid. They comprise prostanoids, leukotrienes, trienes, thromboxanes. Prostaglandins and related compounds are known as eicosinoids. So it is also important if we say classification of eicosinoids. Linolenic acid and arachidonic acid both make eicosanoic acid. After that it makes eicosinoids, and eicosinoids are further of leukotrienes and prostanoids, and these further classified into prostaglandins and thromboxane. What are prostaglandin? It belongs to which group? You all know, eicosinoids. They are present in every tissue in small quantities. They have important physiologic and pharmacologic activities. They are synthesized in vivo by cyclization of arachidonic acid. Food sources of eicosanoic acids are fish oil. Thromboxanes, they are other type of eicosanoids. These are also eicosanoids as we have seen earlier. Discovered in platelets, have cyclopentane ring. Leukotrienes, these are the third group of eicosanoids discovered in leukocytes. They have 3 conjugated double bond. And what are the chemical properties of fatty acid? Esterification, soap formation, hydrogenation, halogenation. What is esterification? Fatty acid form ester with fatty acids. Using esterification process, edible acid, fat and oils can be reacted with edible alcohol to produce useful ingredients, monodiglyceride and lecithin. Soap formation. When fatty acid reacts with alkalis, metallic salt of fatty acid called soaps are formed. Potassium soaps are more water soluble than sodium soap. Hydrogenation. It is a process that where unsaturated fatty acid gets converted to saturated fatty acid. Unsaturated fatty acids react with hydrogen at high temperature and pressure in presence of nickel and unsaturated fat, accept hydrogen and get converted into saturated fatty acid. Means where it's double bond get converted into single bond. So hydrogenation is used to change liquid oil to solid and semi-solid fats. What is halogenation? Fatty acid accept chlorine and iodine at double bond when treated with reagents like iodine, monochloride and fatty halide. What are the chemical properties of neutral fats? There are 4 properties: saponification, hydrolysis, hydrogenation and rinsability. What is saponification? Fat when boiled with alcoholic solution of NaOH and KOH, it undergo hydrolysis. They form glycerol and fatty acid, and fatty acid form soaps with sodium and potassium. What is hydrolysis? Fats when boiled with water at 220 degrees under pressure in autoclave undergo hydrolysis to first form diglyceride, and ultimately glycerol and fatty acid are formed. What is hydrogenesis? process of turning liquid oil into solid fat in presence of nickel and platinum catalyst at 150 to 220 degrees Celsius, and glycerides of unsaturated fatty acid can be saturated by hydrogen partial by hydrogen. So partial hydrogenation produce margarines, shortening partially hydrogenated oils. These products contains trans fatty acid which are detrimental to health as they interfere with biochemical process. Trans fatty acids are more harmful than saturated fatty acids. Very, very important. Rancidity. When unsaturated glyceride oxides or liberation of fatty acid due to hydrolysis, aldehyde forms. This process is called rancidity. In auto-oxidation, oxygen reacts with unsaturated fatty acid and form peroxide that in turn break down into hydrocarbon, ketones, aldehyde and epoxide and alcohol. This results in development of objectionable flavor and color. Condition is called oxidative rancidity.